thank the organizer for the possibility to present this project. So I'm going to tell you about our recent technology to screen biopsy from cancer patients to do drug screening, and how we combine it also with mathematical modeling for personalized therapy. So first of all, what's precision oncology? So essentially it's about matching the right drug to the right patient. And one example where this is really useful is pancreatic cancer. That's the cancer type we are working with in this case. And so it's projected to be the second leading cause of death by 2030. And it has a very low survival rate. So what is very striking is also that this has been unchanged for the past 30 years. And one reason for that is that it's genetically very heterogeneous, and this also is reflected by the fact that patients also respond very differently to drugs. So there is no way to find a drug that works very effectively on all the patients, and there are no way to distinguish responders and non-responders. So it will really benefit from a way to be able to suggest the best treatment for each patient. So the most standard approach to precision medicine is based on genomic, so especially mutations due to the number of sequences of experiments that we have. There are some examples of effective genetic biomarker, but in general, so cancer we know is a very complex disease, so there is no genetic biomarker for most of the drugs. So while not directly measuring the drug effect on the patient sample, the reason is that in general, especially for solid tumors, the number of cells that we can take from the patient is very limited. So either we are constrained to test only few conditions, or we need to do ex vivo culturing. Which is, of course, an opportunity, but it's time-consuming, and it also can change the phenotype of the cells. So what we worked on is a microfluidic platform to do drug screening on patient biopsies. By using microfluidics, we have very small volume, so we miniaturize all the experiment. Therefore, we also need much less cells. So in each droplet, we have about 100 cells. And what we do is to take the biopsy directly from the surgeon, well, from the pathologist, dissociate the tissue, and then encapsulate the cells together with drugs and with a fluorescent substrate. That in our case is caspase-3, so it's a marker of apoptosis. So after letting the drug together with the substrate and the cells, we can measure the activation of apoptosis in response to different drugs. And by combining it with braille display technology, so it's what's used by blind people to read, so it's a series of pins going up and down. We use this to open and close the valves in our microfluidic channel. And in this way, we can generate, it's controlled by a computer, so we can generate in an automatic way plugs with different compositions. So easily combine also different drugs. So each plug that you see here has inside all the cells, it's a 0.5 microliter plug, and it has inside the patient cells and the drugs. So the disadvantage compared to the other method I was mentioning, like organoids, is that we lose the 3D structure of the system, of course. But we can screen directly cells from the patient. So first of all, before starting to screen patients, we actually validated using 2 pancreatic cancer cell lines. So we screen BXPC3 and the ASPC1. So we screen 56 different conditions, so 10 drugs with all the pairwise combinations. And we then look at the conditions that are very cell line specific. So here we rank the top 10 for each cell line. So this is the one that we characterize as very cell line specific. And indeed, to see about the issue of losing the 3D structure, if we can actually recapitulate the in vivo condition, we tested them on cell line derived mouse model. And so this is what I'm showing here. So this one is predicted to be very specific for the BXPC3 cell line, and also in the mouse model, we have a much stronger decrease in the tumor size, specifically for the BXPC3-derived mouse. And what is nice is also that we have a strong improvement with respect to gemcitabine, that's the standard of care in pancreatic cancer. So after testing it on cell lines, we moved to screen patient biopsies. So we are using here 3 different patient biopsies. 2 are from primary tumors. One is a pre-tumor stage, and one is a metastasis, a liver metastasis. We screen the same 56 conditions, and we can see which one are effective in each patient. And in red, you can see the one that the top scored for each patient. So as expected, we see a very heterogeneous sensitivity profile across the patient, and the best combination is different for each patient. So this is nice. So we can, from the phenotype, suggest prioritize at least combination for each patient, but we also want to see if we can gain insight about the mechanisms that are behind this differential response. So what we did is to combine this microfluidic-based data with mathematical modeling. So we start from the prior knowledge on the signaling pathway. We exploit the fact that most of our drugs, so I didn't mention that we screened 10 drugs, 2 are chemotherapy, but all the others are more targeted agents. So we know where they act on the pathway. We have one measurement in the pathway, and we use logic models to generate patient-specific models of the signaling pathway. So for this, we use logic-based ordinary differential equation. I won't go into detail, but essentially it's based on ordinary differential equation, so it's continuous in state and in time. But it derives from the logic rules. So we have also very interpretable parameters. We have one parameter for each that tells us the reactiveness of each species, and one that is the strength of the regulation. And we also do bootstrap to obtain a distribution of the model parameters. So we repeat this procedure for each patient, and in this way we characterize patient-specific logic models. So when we have the patient-specific logic models, we can compare, in this case, between the 2 cell lines, so look at the differentially regulated mechanism. Here we can see that for the BXPC3, we have a strong PI3K AKT pathway, while in the ASPC1, there is a stronger negative feedback loop. And by using ordinary differential equation, we can actually also follow the dynamic of each species over time for all the conditions, so we can also understand the dynamic behind this differential response. Once we have models that are characterized for each cell line in this case, then I will show you also for the patient. We can use it also to do in silico screening, meaning to test different type of perturbation. So we can simulate the effect of different drugs targeting different nodes by inhibiting them artificially in the network, and in this case, in this way, increase the throughput in an artificial way. So these are all our predictions. We have distribution for each prediction because we had the distribution for the parameters. Here again, I'm showing the ones that are specific for the predicted based on the model to be specific for BXPC3 or ASPC1. We start them from the validation of this one in particular because it seems to be relevant for pancreatic cancer. So it's the inhibition of PI3K and the BCL2 family. And we see that our purely model-based prediction can be validated. So we see a stronger response for the ASPC1 and also a stronger synergy. So once we've seen that the pipeline, so we use cell line because on cell lines we can validate. On patients, of course, that's more difficult. So we also characterize the network for the patients, and we can look at the differentially regulated mechanism across patients. So the thickness of the interaction is the strength of the regulation, and the black one are the ones that are differentially regulated between patients. So the rectangle here represents the pairwise comparison, but it's easier to look at it from here. So what we can observe is the clustering, in this case, of the model parameters. And we see that with the model, we better recapitulate also the relationship between patients. So we have that the two primary tumors clustered together, and the most far away one is the liver metastasis. That's also a different tissue, so we expect differences in the signaling pathways. And these differences were not actually evident from the experimental data. And similarly, again, we can use the model also to make predictions for the patients. And in this case, we have among the top 3 that are patient-specific, we have 3 that are in common between the 2 primary tumors, and also based on literature, they seem to have potential clinical relevance. So just to summarize, I introduced both new technology and also how we combine it with the mathematical modeling for precision medicine in cancer. What I didn't mention yet is that one of the advantages is that it allows fast screening, so we actually need only 48 hours after the tumor resection to have the results of the screenings. And it's, the other advantage of microfluidics is that by reducing the volumes, you also reduce the number of reagents that you need, so the experiments become also pretty inexpensive. And the big advantage, I think, is also that we don't need ex vivo culturing. And by combining it with mathematical modeling, we can also look at deregulation and also resistance mechanism, and to generate hypotheses on precision medicine that are also based on mechanism. So just, I would like to thank the people who are involved in the project. So it's the work that I did during my postdoc, shared between Julio Saez Rodriguez lab at EBI, the European Bioinformatics Institute, and Christoph Merten group at the MBL. And these are our clinical partners from the University Hospital in Aachen, and the Matthew Garnett group from the Sanger for the validation experiment. And thank you for this.
Okay, time for quick questions. Any questions? So I just have a question of a very general nature. So I've never worked with the logic-based modeling. It does seem like there's an identifiability issue. So there has to be a certain number of perturbations in order to learn anything. So and also the second part is presumably having some knowledge of the molecular makeup of the tumor mutations, or would inform your modeling too. So just
well, starting from the second question, I totally agree. So is that with the patient material, we are of course limited. So the idea would be to do both and to compare both things. But with patient material, unfortunately, is limited. So in this case, we were more focusing on the implementation of the technology, but I think that for future work, it will be really important also to divide the tissue and to do also more mutational screening. For the first part, so one way to address that we used in the past, we tried also in this case, but it was not really needed, is to use regularization approaches. And the other thing is that by doing bootstrap, we actually obtain a distribution for the parameters. So we also have an idea of the confidence that we have on the parameters that we estimate. And by doing statistical comparisons between distribution, we also address this fact that, of course, not all of the parameters are equally identifiable, but then